Moving on to the inaugural address by Professor Dr. Chandrabhas Rajiv Gandhi Center for Biotechnology. Narayana obtained his PhD in physics from the Indian Institute of Science, Bangalore, in 1995 and carried out postdoctoral research at Cornell University, New York, from 1995 to 1998. He is also an associate professor and dean of research and development at JNCSR, Bangalore. Dr. Narayana leads one of the best interdisciplinary research groups in the country for the use of Raman spectroscopy in drug-protein interaction studies, drug screening methodologies, and for diagnostic applications. He has received national and international recognition for his work. He is also a visiting professor at Pierre and Marie Curie University, Paris, affiliated with the French National Center for Scientific Research. He has several patents and about 100 heavily cited papers in high-impact international journals. On that very brief note, I warmly invite Dr. Chandrabhas Narayana to deliver the inaugural address.
Thank you very much for the kind introduction, and I would definitely like to thank Professor G.M. Nair and Kerala Academy of Sciences for giving me this opportunity. Actually, it is an honor to be invited, and I consider this body as an eminent scientist conglomeration, which actually brings together all of the Kerala Malayali or Keralites into this academic fore. But it is a wonderful initiative also from the big stalwarts in Kerala for setting up such a academy. So to begin with, I would like to acknowledge the presence of our eminent people here. So Professor G.M. Nair, President of Kerala Academy of Sciences, previous former presidents, Professor Umman V. Umman and Professor P.R. Sudhakaran. Then I would like to thank KB Ramesh as General Secretary KS, who actually was coordinating with me for this program, as well as the Vice President, Dr. N.S. Pradeep. I also would like to thank Professor Unnikrishnan Nair for coming over to listen to me in the morning here. And I like to thank all the audience who are eminent scientists as well as students and postdocs, ladies and gentlemen. I'm going to actually give you a new tool which is coming up as a possible applications in biotechnology, and that is one of our only Nobel Prize winning technique. So I'm really thrilled to share some of the latest work which I have been carrying out for this webinar as a part of the inaugural speech of this webinar, to motivate people to see this new technique probably may be very useful in phytochemistry also. So I have chosen some examples which are my latest work, which relates at least to some of these molecules which come out of for medical applications here. Let me share my slide. Yes. I hope you are able to see my slide.
Yes.
Yeah.
Okay, so I have titled my talk as Raman, a new baby in the block for applications in biotechnology, because Raman was never thinking that this would actually will be a useful tool for biology when he discovered Raman effect. And considering the audience to be mixed, I would like to start from the basics so that people can understand why Raman is useful in doing these wonderful experiments in biotechnology. So to begin with, it is a inelastic light scattering process. And when I say scattering, so you have both elastic scattering and inelastic scattering. And when I say elastic, that means the scattering contains the same color of the light which falls on it. And this is an example of elastic scattering from dust particle. So let me take the laser pointer. Yeah, so this is the elastic scattering coming from the dust particles in the room, and this is the elastic scattering coming from the liquid, which is colorless. And if you look at it, it is cyclohexane, and it is very intense. And because it is intense, it saturates your detector, and it is in all directions it is coming. But along with that, inelastic scattering means change in color. It can be red shifted or blue shifted. So if I want to show you right here, I just mask the blue light with a very good filter nowadays available. And you can see the disappearance of the blue color here, which was present earlier from the dust scattering. And all of a sudden, you start seeing a shifted frequency, yellowish green color coming from this liquid. And this is the Raman scattering, which Raman discovered. And he was working with mostly organic molecules. And they give you very strong signatures in Raman. So Raman had used a simple prism to disperse that light which was coming, and he would take a photograph. And this is the first Raman spectrometer, which is present in Indian Association for Cultivation of Science. And this was the one which gave him the Nobel Prize. And this is the first photograph of the Raman scattering, which is red shifted is here, blue shifted is here, and this is the elastic scattering, which is very, very intense compared to the inelastic scattering of light. And this is the one which got Nobel Prize. Today, we can actually take a laser, which is like your laser pointer, and it is available in Amazon for 1,000 rupees, green laser. You can put a filter to filter out and make it monochromatic, and then put it onto a liquid or any sample which you want to study. And then you put a filter to filter out the green light and take it through a fiber and disperse it using a grating. And you can use an SLR camera, and you can take a photograph. And you can see the elastic scattering and the red shifted light, which is coming. And this is the Raman scattering, which you are actually seeing. And if you digitize it, you can actually see for each of these lines, there will be a peak associated with it. And so this is basically how we can do and how simple Raman spectrometers can become from what people nowadays use, very commercial ones. These shifted light is because of the vibrations of the molecule, and they are very unique. Any diatomic molecule would have only one vibration associated with it, which is symmetric stretching. And if you see, I have selected HF and HCl, which are basically difference is fluorine and chlorine. And you know that chlorine is heavier than fluorine. And the heavier they are having lesser energy compared to the lighter ones. So it also depends on the bond strength of the diatomic molecule. And this is why they are very, like a fingerprint technique. And the moment you increase the number of atoms, you would have increase in the number of vibrations. And here I have taken water molecule. It is a AB2 kind of a molecule, which is a bent molecule. And you have symmetric stretching, asymmetric stretching, and symmetric bending. And you can see it is easier to bend a bond compared to bend a bond angle, whereas here it is stretching of the bonds. So they are more energetic compared to bending. So this is how you can understand. Now, all molecules which look like AB2 will have a similar stretching and bending modes, but their values will depend on the constituent atoms as well as the bond stiffness of it. Now, I have taken another example here, CH2, which looks similar to water. But so it does have symmetric stretching, asymmetric stretching, and symmetric bending. But because CH2 has two R groups attached to it, like a bulky group attached to it, that gives it an additional vibrations associated with it. This is the twisting, this is the wagging, this is your fanning kind of motions, which also happen to come across. So depending on the type of the molecule, you can actually see that the vibrations are associated, and they are like unique signatures. And if you have an environmental change, that vibrations would be affected, and that actually is the reason why this becomes a very important tool in diagnostics. So as I showed you earlier, the shifted frequencies are seen as peaks in your spectra. And these peaks, all of these peaks can be associated with vibration. And in this case, we are looking at carbon tetrachloride, which is a tetrahedral molecule, AB4 type of molecule. And any AB4 molecule will have a similar spectra associated with it, except for the energies will be different. This is symmetric stretching, asymmetric bending, and symmetric bending. So you can see the bending always lower frequency compared to the stretching. Now, you have computer programs which can actually tell you what kind of vibrations are associated with any set of atoms connected together. And that is the reason why now Raman has become very powerful in looking at molecules itself. But if you look at it very carefully, the symmetric stretching of this molecule is not definitely looking like a single peak. So whenever you have in spectroscopy a possibility to see multiplicity of peaks, you just lower the temperature, because when you lower the temperature, the width of the peak actually reduces. And this width reduces, so suddenly you see it is not one peak, but it has actually 5 peaks. And these 5 peaks are because of the fact that the chlorine is having an isotope which is 35 and 37. And I told you the peak depends on the mass of the atoms also. So 37 is heavier than 35. So naturally, these are the reason why we have. So when you look at it, I've just put it across in the inverted way. So the lightest atoms would give you higher frequency, and the heavier atoms will give you the lower frequency. So you can see that all Cl34s, Cl35s are there, then it will be the highest frequency. And you can start seeing that it will actually be increase in 1 Cl37, 2 Cl37. And obviously, you can now guess that what should be the reason why this is there. And also, you can actually know that the weakness in the band is because of the relative abundance of that. So the tallest peak is 3:1, which is your relative abundance of the chlorine atom itself. We have been building Raman spectrometers, and some of my patents are based on that. This is one which I have actually made and given to ICER Trivandrum for pesticide detection. This is a setup which is there. And you can see you can also put a 96 well plate, and you can do a scanning. So you can do a 96 samples at one shot, and you can record the Raman spectra. Now, to show you that my Raman spectrometer and a commercial Raman spectrometer here. So the green graphs are all commercial setup, and you can see the setup which we have built. You can see one-to-one correspondence between the molecules which we have recorded. And you can also see the power which I am using is similar, and the time taken for acquisition is much lesser. But of course, because I have miniaturized it, it actually has a lower resolution compared to the higher spectrometer. But you can see that we have very good throughput in our system, and it is very, very comparable, and its cost is like 1/5 of the cost of the commercial setup. So you can actually do research grade work also in such systems. But Raman is a very, very weak phenomena, unlike absorption. So IR is an absorption technique which is complementary to Raman. So people have been always looking out how do we increase the Raman signal. So this was in late 90s that people realized that if you take gold or silver nanoparticles and if you put the molecule on top of it, and if you incident the light, all of a sudden the molecule actually gives very strong signatures. This phenomena was called as surface-enhanced Raman scattering. And it is because of the fact that you will actually develop a dipole which is oscillating because of the electric field itself, which is oscillating, and that dipole actually produces any oscillating dipole produces electromagnetic radiation. So the molecule actually experiences not only the electromagnetic radiation coming from the light, but also electromagnetic radiations coming from the nanoparticle itself. And for this, you should have enough electrons in the system, and that's why the gold and silver become very good material, because they are the best conductors available in the elements. So this is the reason why they give you a very strong Raman signal. So using this, we can actually do a lot of stuff in biology. And I am going to show you that I am using this as a screening tool for finding out new inhibitors. So if you have a molecule and you want to pre-select them, that this is the molecule which we should do the biochemistry and try to find out whether they are much better for inhibitors or activators, then this search technique could be a very good screening tool for it. I'm looking at JNK kinases in this case, and I was working with Guru Rao, who is a chemist in IISc in the solid state structural chemistry unit. He's a crystallographer by training, and my student Shantanu and others were involved in doing this work. So as we know that kinases are basically the ones which take an ATP, convert into an ADP, and phosphorylate the protein. So the protein phosphorylation is done by the kinases. And the name nomenclature generally comes from the fact that the protein which is being phosphorylated is the one which gives the initial name. And this is the c-Jun protein, and its N-terminal is where the phosphorylation happens. So they are called JNK, and there are three of them, JNK1, 2, and 3. And these are very important kinases, because any external stress on the cell stimulates JNK secretion itself. So any abnormality in the phosphorylation of the c-Jun is a potential threat. It actually gives you neurodegenerative diseases, chronic inflammation, birth defects, cancer, ischemia, and injury, obesity, and insulin-resistant diabetes. So all of these things are actually because of the c-Jun's abnormal phosphorylations. So it is very important that when you have such an abnormality, you should be able to inhibit this phosphorylation of the c-Jun by JNK. And inhibition can be in two ways done. One is by this is your substrate which actually binds to the enzyme, and your active site is here. So the inhibitor could be a competitive one where it competes with your substrate, or it can be a non-competitive one where it attaches to the surface of the protein, changing the active site so that the substrate doesn't bind. So both of these things are possible, and that is like an on-off switch which we can actually work in the system. So in the case of c-Jun N-terminal kinase, the protein crystallography was done, and they have also understood that this particular molecule, which is pyrazoloanthrone, which is a very good inhibitor for this system, but not as great to become a medical applications in the system. So this molecule was known to Guru Rao's group, and they were trying to find out better derivatives of this molecule so that they can actually get a better inhibitor from these molecules. So they realized that placement of chlorine atom at 7th position, that is this position, causes a good twofold improvement in the inhibition from their inhibition studies. But they were not able to understand why, and they were actually doing serendipitous experiments where they are doing known derivatization of the molecule. So some of the molecules they were looking at is they actually made an amine attachment to this group here, and they also played around with the position of the chlorine. And all of you should realize that chlorine is a hydrophobic pocket, and this is a hydrophilic region. So this is a playing of the hydrophobic hydrophilic region in this system. They also tried to make it with a sulfate group, and they again did the similar experiments in this one, and they also got an azo group attached to it and tried to do a modification to this there. So they had 9 derivatives, and they wanted to know how does this actually work, and what is the reason why we can actually see one of them is better than the other. So usual technique for derivatization is that you synthesize new molecules, do the biochemical studies for all the molecules, then study mouse models for it, and then try even do an X-ray crystallographic study to understand where it is binding, and MD simulations to understand what is the kind of interactions taking place. But if we can have an alternative approach that you synthesize new molecules, and if you can pre-select it by search technique using a screening tool, so the number of biochemical studies on molecules can be reduced, and you will only look at the screened molecules, and then you can do most of the other things. So it actually reduces the number of molecules you produce, or you can at least get a hint of what kind of inhibitions can be done. So this is the question where we were actually interested in. So can search be a very good molecular level information for the inhibitor binding to the protein? Can we differentiate between the inhibitor binding of one protein to another? And can we predict which is the best binder for this system? So this is what we actually were working, because the X-ray crystal data was there for JNK1 and JNK3. We could do MD simulations of this also. So when we do the MD simulation, we can see by structural superimposition, most of the structure are very identical. And you can see that we have done an electrostatic potential mapping of this. This is very important. Later slide, I will tell you why. And then you can also look at the hydrophobic pockets in the protein itself. And now if you look at the primary sequence, you can realize that there is a 92% sequence identity present. And one of the problems when you have such a good sequence specificity, that means if an inhibitor works for JNK1, it will also work on JNK3. That means you will be actually not getting the desired result. So we should be having specific inhibitors for JNK1 and JNK3 and try to do this. So that is one of the challenges when we are working with related structures of the protein. Now, when I was talking about electrostatic potential, because the silver nanoparticle itself is charged, it has a negative charge, so it will actually attract the protein in the regions where there is a large protein positive charge present. And from the electrostatic map, we realize that arginine is the region in the protein. It's a dimer. So it's a dimer, and you can see that arginine would be the anchoring point in the case of JNK1 in the system. And also in the case of JNK3, the anchoring points are lysine and arginine 50. So there is a difference in the way these two proteins would be orienting itself. But one of the interesting aspect is that the active site, which is where the ATP binding happens, ATP conversion happens, this region is within a few tens of angstroms. That means any molecule which binds here would be immediately able to give you a very good search. Now, I wanted to tell you that most of the molecules which we have produced are insoluble in water, and the silver nanoparticles are present in aqueous solution. So naturally, we can never get a search of the molecule itself by default. So if they are anchored on the protein, only then we would get the signature of the molecule. So this is a very good understanding to know whether the binding has happened in the system itself. So just to show you that if two similar looking proteins are there, the Raman spectra would also be very, very similar. So this is JNK1 and JNK2. There are very subtle variations which are happening, but most of it looks very, very similar to each other. So only a trained person can distinguish between the two proteins, but you can see that similarities actually are reflected in Raman spectra also. Now, before I go forward, since the molecules are binding in these active sites, and these molecules are organic molecules, and I showed you that organic molecules give you very strong Raman signal. So the protein signatures are masked by the Raman signal of the molecule if they are bound. So what we realized is that we have taken all the molecules and looked at the Raman spectra, and we can see that it is a convolution of the Raman spectra of the molecule and the protein itself. So there are some similar peaks, but some are actually getting modified. But the most important thing is that the intensity of the Raman signal. Intensity of the Raman signal is given by these numbers, and you can realize that the strongest signal comes from RAN2, which is basically the one which binds much more stronger compared to the others. So if you plot it as the counts per second to the better binding, you realize that there is a one-to-one correspondence between the binding studies and the intensity of the peaks. And this is the order in which the binding is increasing. And you can see the parent molecule is actually much weaker binding to JNK1 compared to the RAN2. And this is the modification which happens in RAN2. So the chlorine present in the 7th position and amine in the as a derivative in this side is the one which is the best for JNK1. Let us look at JNK3. Again, we can see that the molecules are actually binding differently. And of course, you can see SP is much better than most of the molecules which were there for JNK3. But you can see RAN2 still is much better binding. And you can see the intensity ratios are much larger. You can see this is 160, 182. So it is a much better binding molecule. So Raman can immediately tell you which molecules you should be looking at from this system. Now, the next thing which I wanted you to see is that the binding of RAN2 on JNK1 and JNK3 are not the same. So if you look at the Raman signature, you see most of the Raman peaks remain similar. There is no change in the molecular signature, but you see a very strong change in the Raman intensities. And this is very important because this is very much dependent on the polarizability of the molecule. So the way the molecule is oriented with respect to the nanoparticle will give you different intensities. And now we realize that this peak is very much dependent on how this molecule is oriented. So if you see in one case, in JNK, it is having an orientation in this order. And in the case of JNK3, it is in this fashion. And that change in this is causing you to have the intensity change. And we have done the molecular dynamic simulations, and we do see that in the pocket, active pocket where it is binding, we can also see the groups, amino groups, which are actually binding in that region. And its orientation is different. You can see here chlorine is facing towards you, whereas amine is facing towards you in this case. So this is the flip, which is also being very easily seen from the Raman spectrum. So what we have shown here is that it can be a very good screening tool, and we could actually show why JNK1 and JNK3 are inhibited better by RAN2 because of the binding being very strong. And you can use SIRS as a quick drug discovery tool because it shortens the number of molecules you will be working in the derivatization of the protein. This is not the first time. This is our latest work, but we have done this and published many before to show that we are able to show the binding as well as the screening of the tools. Now, in the last part of my talk, I am actually showing you something where I am actually working with a company, Biodecipher in Bangalore, which is actually having MD as Jayaprakash Sargoli, who has been working with me for many years now to find out Raman as a very good tool for detection of indels in bioindustry. That is very important, and we are using search probes to actually study this. Now, when we talk about the mutations in the plant region, there are many mutations which are called the single nucleotide polymorphism, which happens, but also sometimes deletions happen as well as insertions happen of one or more nucleotides in the system. So here is an example of SNPs where A is being replaced by C and A is being replaced by T, or G is replaced by A in this, that is called the single nucleotide polymorphism. Or you can have completely missing nucleotides in the system, which is called the deletion. And if you have an insertion of a nucleotide, it's called insertion. And generally, this insertion deletion is called the indels in the system. Now, you know that the mutations actually cause a lot of changes in the appearance. You can see the change in the appearance of the systems. Growth rates can be affected, the height of the plant, yield, as well as susceptibility to the diseases can be also modulated by these mutations. And this is very importantly in the biotechnology area, in the plant biotechnology, where we are actually looking at yield and disease resistance in the system, and we are trying to look for that. So it's very important that we detect these SNPs in the system, and normally used techniques are gel-based method like tetra-arms PCR, or non-gel based methods like CASP. So competitive system, which is very, very important to do. Here, gel-based methods generally are PCR followed by a gel electrophoresis, whereas non-gel-based are fluorescent dye-tagged DNA sequences. So these techniques are being used, and my collaborator in the industry has been using these for detecting it, but he found that we could actually minimize the cost, make it portable by using Raman techniques after he has seen my many of the inventions which I have done in the lab. So we actually have seen that people have realized that we can even do DNA sequence detection using search. So basically, here is an example where we can see that if suppose a particular sequence is there, we can actually detect that sequence by putting a complementary strand and bind it. And this is something which people have done. And you can see the differences in the intensity, because if suppose no template exists or negative control is there, you will see the intensity of the peaks being increased only when the sequence specific attachment happens. So this is something which is useful in doing these kinds of studies of indel. We were actually looking, and especially in my collaborator was, one of the problems he was looking at is the varieties which are which can be resistant to tomato yellow leaf curl virus. This is something which he was looking at, and this is a very big problem for the tomato crop, because it actually causes virtually eliminating fruit production in the system. And so it is being spread by this silver leaf white fly, and you can see the signatures of it by marginal leaf yellowing, reduction of leaf size, flower and fruit drop, and plant shunting. So all of these things give you an idea that this particular virus has actually affected the tomato crop. Now, we realize that if suppose this sequence exists in this plant, tomato plant, then it is resistant to this particular virus. And so it is necessary to find this sequence which has been inserted in this genome of the tomato. And we are interested in seeing whether we can use search for it. So we have used FAM dye as the Raman marker for the presence, and Psi3 tag for absence of this system. So we do a limited PCR of this system. So we have selected resistant indel for the resistant part with a FAM attachment, and this is your probe. And for detecting indel deletion, that is non-availability, which means it is susceptible for the disease in the allele, and that probe has been designed. And here is the forward primer and forward primers which have been made, and we have got it from IDT, and we also get it from Eurofins, these probes. The difference is IDT actually does a half indel sequence, while Eurofins do full indel sequence. So that is the only difference between the two. And we wanted to test out both of them. Now, when you look at the FAM dye and Psi3 Raman spectra, they are dominated by fluorescence of these materials. And this is without any DNA in the system. So you can see that this is the Raman spectra of FAM, and this is the Raman spectra of the Psi3 attached to your probe molecule. We actually look at a particular vibration in the system which is unaffected by the orientation of the molecule on top of the your nanoparticle. So we have identified these signatures, and that is what we will be actually monitoring when we have whether negative control or a positive control, and we will actually look at these things. Now, this is a indel deletion detection using IDT primers, and you can see when there is no DNA added, you can get a signal coming from the system. So this is to test whether the probe is present in your PCR equation. So that gives you a signature. But when you put a negative control, that means the DNA is not the sequence which is supposed to be present in the tomato, then you get a negative control which is clustered around a very low intensity. And when we have this sequence, then it gives you a strong signature around it. So there is a clear distinction between the presence of the sequence and any sequence which is not having that. So you can actually detect it very easily. Similarly, we did it with the full Eurofin primers, and you can see a similar effect happening in the system. Here, this is the control. So where we do not put any DNA, you can still see that the Raman signatures come. Now, the question you may have in the mind, how do you detect it? Because as long as you put any DNA into the system, this signature will never come in the system. So any presence of an oligonucleotide or any of those things will actually kill the signature. So that is why it will be a low intensity. And the moment you have a very specific attachment of the signature, it will actually give you the signal. And so this can be easily detected. Now, to show you the Psi3 also works in the system, we can see the exact same, that when you do not have the, deletion is not there, then it actually gives you a lower intensity compared to the one which is higher intensity. And you can see the intensity difference is factor of 2. And that means you can easily detect these systems in these things. With that, I would actually try to show you, thank you for your attention. And what I wanted you to realize is that this is a technique which actually can be used in multiple ways. I have only shown you 2 different examples of how this actually helps you in doing many things. The previous last example I have shown reduces the cost of the detection by a huge amount, because when you do a PCR, you need a quencher as well as a fluorescent dye. And the quencher and the fluorescent dye attachment itself is a very expensive proposition. We have removed one of them. Second thing is the PCR cycles we do is very less. And now I also wanted to emphasize that the last experiment were done with picomolar of DNA. And you know, the plant DNA extraction is very tough. So you really don't need to do a large amount of sample detection so that you can get a very good detection of the system. So we can work with very low concentration of the DNA. We can actually do with the limited PCR, and we can actually also look at what is happening when you have a deletion or when you have an insertion in the system. You can clearly distinguish these easily. Thank you very much. I will stop here, and I'm happy to answer any questions if the time permits. I hope I was in time.